Hello everyone, so we're back for a brief while and I will do a quick review of everything that we've seen in the first evaluation. First we started with the cell. If you remember, the cell had several compartments that are called organelles. Okay, we did one of the tasks where you had to draw a cell, okay, quite like this, with at least 14 organelles. Now we're going to see which organelles are the most important. These things that are that I just drawn are outside molecules of the cell. Okay, they're called cell markers. We're going to use them in future evaluations, but not really this one. So the cell has several important compartments. One of the most important ones is called the nucleus, as you can see here. Then we have the endoplasmic reticulum outside the nucleus. It's a system quite like this, with several cisternaean tubula, and then we have the Golgi apparatus that is quite the same as the endoplasmic reticulum, but a little bit more independent. Okay, so the Golgi apparatus is like the final stop for everything that comes out of the cell. Okay, the endoplasmic reticulum, well, this is the Golgi apparatus or Golgi body. Okay, both names are correct. Then we have the endoplasmic reticulum. Endoplasmic reticulum that can be rough or smooth. Okay, in the exam you cannot abbreviate endoplasmic reticulum. You have to write it. Okay, the rough endoplasmic reticulum, you can distinguish it from the smooth because it contains attached to the sides ribosomes. Okay, like this. As long as it has ribosomes, it will be the rough endoplasmic reticulum. And then we have our friend here inside the nucleus. I didn't put nucleus here. Okay, inside the nucleus we have our friend, the genetic material. It is going to be the nucleolus with this big blob of ink, and then DNA, like this. Okay. Nucleolus, and then we have DNA. A little bit crowded, but that's what we have. Then we're also going to have mitochondria that gives energy to the cell, and we're going to have, last but not least, we're going to have, lysosomes and endosomes and all different membrane things, and free-floating ribosomes all along the cell, like this. So that's like everything in the cell. You have a PowerPoint presentation where you can see all of this with detail. This is like a brief sum up of everything. Now, out of the inside the nucleus we have DNA. Okay, DNA, we've studied the levels of organization. Then I need to do also a quick sum up. We have the DNA double helix that has the basic unit is the nucleotide Yes, you remember the nucleotide that has the nitrogen base, has the deoxy ribose and the phosphate group here, okay, and nitrogen bases can be A, T, C, and G, okay, A pairs with T, T with A, C with G, and G with C. And the sequence of nitrogen bases is what makes up the genetic code, okay, super important. Sequence of nitrogen bases equals genetic code. Now, if you remember, okay, the DNA double helix is very weak. Oops, sorry for this clumsy DNA double helix. So the DNA double helix is something that is very weak. So we have to protect it. Yes? How do we protect it? From now I'm going to draw it as a line. Then we're going to protect it with the histones. Okay? Histones are going to be in purple. Okay? So we're going to have histones in the DNA like this. Well, histones in the nucleus, sorry, like this. Okay, and the DNA is going to wrap itself up 2 times around every histone, forming what we know as the nucleosomes. Okay, nucleosome is all of this. Nucleosome. Several nucleosomes together, okay, more than one is the per nucleus structure. Now, if we start coiling this thing up, okay, now this is going to be drawn as a line. So if this line starts coiling up like this, okay, with all the nucleosomes all together, it's going to form chromatin, okay, and if the chromatin super condenses or coils, okay, it's going to form chromosomes. Now we're going to see a little bit more about the chromosome structure, okay, and for that we're going to change colors again. Well, no, we're not going to change colors. So for that we're going to see the chromosomes. Okay, so chromosomes, most common representation of a chromosome is something like this, yes, as you already know. Okay, so chromosomes have lots and lots of nitrogen bases, okay, millions of them, and has several different structures, as you know already, because if you don't know and you have an exam next Wednesday, then, well, you should rethink how you study biology. So this is a chromosome, okay? A chromosome. So the chromosome has structures at the end here, 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 and here. They're called telomeres. Then it has another structure here at the center. This is called centromere. Then we have arms, okay? This is an arm. The upper arms are going to be the P arms, and the lower arms are going to be the Q arms. So then we have another superstructure in a chromosome. That is a chromatid. Okay, so this chromosome has two chromatids. Okay, you can see one here and another here. Super important. Okay, this is a chromosome that has twice the normal information, because chromosomes normally have only one chromatid. Okay, so there are several types of chromosomes. That we haven't seen, but I need to be super quick to explain. So the types of chromosomes okay, as you've seen in the lab practice, there are chromosomes that have the centromere low in a normal position, or they have it just here upwards. So they're going to be divided in several types. Okay, the first type is the metacentric chromosome. Metacentric chromosome is going to be like this. Both arms are going to be equally distant. Okay, they're going to have the same length. Then we have the submetacentric chromosome that has the upper arms a little bit shorter, okay, than the lower arms. You can see obviously that these are quite shorter than this submetacentric chromosome. Then we're going to have the acrocentric chromosome that is going to have super little upper arms, okay, like this. So the P arm and the Q arm, okay, the P is going to be always the shortest one. And then we're going to have the telocentric chromosomes that have a little thing here. Telocentric because of the telomeres, okay? Telomeres, they indicate the end of the chromosome, okay? The very end of the chromosome. And also, super important, are the genes, okay? Remember that a gene is a bit that contains the information to code for something, okay? For example, the gene for the color of your eyes, the gene for the color of your hair, or the shape of your hair, or your height, or how many fingers do you have, okay? The gene here. And the genes have super special positions depending on where they are, and they're going to stay in the same chromosome, in the same place, for the same species, if you remember. And that's going to be called the locus. The locus is the place of the gene, and remember that it has the chromosome number, the arm, and the distance to the centromere. Okay, distance to the centromere, arm, okay, and the number of the chromosome. Last but not least, remember that we have 23 pairs of chromosomes in humans, okay, and that there are mutations that affect the structure, the shape of the chromosome, and the structure. Okay, we can see really super quick the mutations here in this purple cloud. Okay, mutations, remember that we have numerical mutations are trisomy with one extra chromosome and monosomies that have one less chromosome. And then we have structural mutations. We have duplications. We have deletions. We have translocations. We have rings. And we have inversions. Remember that you have everything in a PowerPoint presentation where that you can consult and see everything. Now we're going to get a quick review of mitosis and meiosis. So pay a lot of attention, really, not just a little bit, and you're going to get the actual PowerPoint presentations from your classmates as soon as I can correct them. Okay, so pay attention for the second part of the video, that is going to be mitosis and meiosis. Okay, so now we're going to get to the mitosis and meiosis. Okay, so how does it all start? Okay, first we start, well, this is called, really, this is not mitosis and meiosis, this is the cell cycle. Okay, the cell cycle starts with something that is called interphase. Interphase is the biggest part of the cell cycle, and it has several phases. First, we start in phase D1. In phase D1, the cell starts replicating things. Okay, all the organelles, in order to make another cell, we need to have two of everything, because if not, the new cell is going to have only half of the things, and we need to have everything. Okay, so the first part is going to be the interphase, where it replicates most of the stuff, mainly organelles. Then we're going to get the S phase. Okay, the S phase, where it's going to replicate DNA. Then we're going to have D2. There is a phase that is called the double check, where we're going to see if everything is okay. We have two of every organ, two of every thing, okay? And then we're going to get into the mitotic or meiotic phase, okay? And here, at the very end, when we have two cells, okay, we can have something that is called G0, where the cell stops replicating. For example, neurons, okay? Neurons don't keep on replicating all the time, so they stay in G0. So G1, all the organelles, S, the DNA and other genetic material. G2 is going to be a double check that G1 and S have gone right. And then M, that is the mitotic or meiotic phase, where the actual replication is going to take place. Okay, the actual replication and organization of the things is going to take place, and that is the thing that we're going to explain right now. So, we're going to start with the easiest one, okay? The mitosis. Mitosis. So in the mitosis, we start in the M phase, okay, with a very big cell. Yes, that very big cell is going to have a very big nucleus full of lots and lots and lots and lots of DNA. Okay? Yes, that is the start. We're going to have a lot of organelles and blah 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 blah. Okay, so in the first step, we have the prophase. In the prophase, our very big cell, okay, is going to do several things. The first one is that the nucleus is going to disappear. And the second one is that we're going to start forming chromosomes. Okay? In this case, okay, 2n is equal to 2. 2n means that our cell is diploid, okay? That means that we have 2 times the normal amount of chromosomes, okay? So we have, remember that all our chromosomes are set up in pairs, so we're going to have 2 pairs of chromosomes. Okay, we're going to have 2 of every chromosome, so it's in pairs. Okay, so we're going to have a total of 46. So 2n is equal to 46 in humans, and in our sample cell 2n is going to be equal to 2. So in the prophase, chromosomes start appearing, and then another structure that is called the centrioles, okay, is going to appear also. Okay, let me draw them here. These are centrioles. Then we're going to have the metaphase. In the metaphase, this is a really quick gathering of the mitosis, okay? There will be detailed PowerPoint presentations with every phase in the near future. So in the metaphase, okay, and the chromosomes are going to align in the center of the cell, thanks to our friends, the centrioles here, that are going to extend microtubules, and they're going to be attached to the centromere, okay, like this. So if this is a chromosome, then the microtubules are going to attach here, like this, and that structure in the middle This thing is going to be called the kinetochore. Kinetochore, it means kinesis, that is moving, and core, that is the nucleus. So it's the nucleus of moving the chromosome in this case. Okay. In the third phase, that is called the anaphase, the cell is going to become elongated like this. Okay, you see that it is stretched. That is thanks to other types of microtubules. Okay, they are going to push against each other like this. Okay, in order to elongate the cell. And in order to keep the cell in place, we're going to have yet another kind of microtubule. Okay, those are called astral and polar microtubules. The astral are going to be the ones that push against each other in order to make it longer, and the polar microtubules are going to be in the poles, and they're going to keep the actual shape of a cell. Okay, so anaphase is super simple. Okay, the chromosomes are going to break in the middle. Okay, and then they're going to move to the opposite poles, helped by the microtubules. Okay, microtubules are going to be the things that move the actual chromosomes. There are thousands of microtubules in a cell, not just 3 or 4. So the microtubules are going to, if this is a chromosome, okay, with 2 chromatids, it's going to break, and then it's going to be pulled apart. It's going to move to the poles. Okay, they're going to move to the poles, and then we have the telophase. We have, the telophase, where the cell is fully elongated. Okay, the genetic material is going to be at the poles. Okay, and then the nucleus is going to reappear. But we're going to have 2 nucleuses, 2 nuclei. Sorry. The nuclei, okay, are going to store the genetic material as the chromosomes become chromatin again, okay, and disappear. Then the microtubules are going to stay here, okay, keeping the shape, and in the middle there's going to be nothing. All the organelles, all the organelles are going to move also, okay? In the metaphase, in the metaphasic plate, the thing in the middle where the chromosomes align, okay? This that I'm going to, this is the metaphasic plate. Metaphasic plate, okay, that is going to be the structure, the imaginary plane in the middle where the chromosomes align, okay. There's not going to be organelles in the metaphasic plate, okay, they're going to be at the sides of the cell, pulled by the microtubules. In the anaphase, the organelles are also at the side, okay, here at the sustained by the polar microtubules, and here in the telophase there's going to be nothing, okay, in the middle, just like microtubules like this, because there's the last phase that is not really a mitotic phase, but for easier explanation, I'm not going to say anything else. Whoopsie. That is the cytokinesis. In the cytokinesis, the elongated cell is going to become thinner in the middle, okay, with the nucleus, and it's going to become thinner and then it's going to divide. Okay, and this is called, this structure here is called the cleavage furrow. Okay, so this is mitosis. Now we're going to get to meiosis really quick, and that would be the end and everything that is going to be in the exam. Okay, so here you have a nice sum up of the mitosis. Okay, remember in the prophase, nucleus disappear, the chromosome forms. Okay, and the centriole migrate to the poles, creating the spindle microtubules. Okay, spindle microtubules. Then we have the metaphase, where the chromosomes align in the middle. That is called the metaphasic plate, and the kinetochore forms. Okay, that is the microtubules plus and the centrosome. And in the anaphase, the chromosomes are pulled to the poles by the microtubules, and in the telophase, two nuclei appear in its pole because the chromosomes are already there, and the chromosomes melt sort of or uncoil into chromatin inside the nucleus, obviously. Okay, and then we have the cytokinesis where the cell divides in two. Okay, so now we have the meiosis. Okay, meiosis is special. Meiosis is special because it's going to be the guilty part on something that we call the genetic difference. Okay, we are different from our parents because we have something that is called the sexual reproduction, and you know that you get half your information from daddy and half your information from mommy. But thanks to the meiosis, okay, we're going to have a special process that is called the crossing over, where the information from the parents of your fathers is going to mix. Okay, and I'm going to explain how. In the meiosis we have two divisions. Okay, let's start by that. Why? Because the meiosis is going to be guilty for creating the sexual cells, okay, the gametes. So in the first division, okay, we have a cell that is 2N diploid, okay, so it has two copies of your genetic information. Okay, that is where the chromosomes are divided in pairs like this. Okay, we're going to have a pair of chromosomes. Then when you pass through all the phases, okay, normally in mitosis you're going to have another 2N cell. Okay, so this 2N cell is going to get you another 2N and 2N cells. But in meiosis, okay, there's not another interface between divisions. We get to the next division, okay, really super quick. So if we don't have an S phase where we duplicate the genetic material, okay, and we divide 2n by 2, we're going to have cells, okay, that have only n information. Okay, n is called haploid, okay, and have the information. So here we have the chromosomes like this, okay, only chromatids. So here we have just one chromosome that is one chromatid. Okay, so we've divided the information like this. So 2n is diploid. In our case, is 2 times 23 chromosomes, and n is haploid, that is going to have only 23 chromosomes. So 23 plus 23, when we meet, plus another, is going to have 46, that is 2 pairs of 23 a pair of 23 chromosomes for every cell. So you have a total of 46. Okay, therefore you have a full human being using the gametes from 2 adults human beings. Okay, that's how you have babies. Now in the prophase, okay, in meiotic prophase here, in this one, okay, we're going to have a special thing that is called the crossing over. Prophase of the first meiotic division. Okay, we're going to have, you have, okay, a normal human being has a chromosome that comes from the father, okay, like this, and a chromosome that comes from the mother. Like this. Okay, so when in prophase 1, they're going to get really close. Okay, this is step 1. In step 2, they're going to get super close. Okay, this is obviously the same chromosome. Okay, this is one pair of chromosome, this is one pair of chromosome. Okay, they're more or less the same, they have more or less the same size and more or less the same everything. Okay, so just bear with me, these are the same. Okay, so they're going to get super close, one of the chromatids. Okay, like this, and they're going to become momentarily joined like a zipper. This is called a tetrad. Okay, in step number 3, one of these close points, okay, randomly, okay, is going to cross over to the other side. I'm going to make them bigger so you can see. Okay, so it's going to sort of cross over to the other side. Remember that they are super close. Like this. And then they're going to break off. Okay. And this is the really spectacular thing. Okay, we're going to have a bit of your mom's chromosome in your dad's chromosome. Okay, and a bit of your dad's chromosomes in your mom's chromosomes. Okay, like that. And finally, they're going to become detached. Okay. So what just happened here? Okay. Super easy. You got a mix of things. Okay. So a chunk of information has crossed over to the other side. That's why you can have the eyes of your dad and the skin of your mom. Okay. Oh, it looks like your dad, but it's a little bit like his mom. Okay. That's how you get that, because the information mixes, the information crosses over. A little bit of science. This thing is called the chiasma. Okay, the actual point where they cross over is called the chiasma, and this process is called the crossing over. Okay, and that's how you get a mix of information in the new human being. So it doesn't really quite looks like the dad of the mom. But this is not just looks. Okay, this is the very basic thing of evolution. Okay, evolution is based on this, because that way we can get the best thing from two individuals, the better things from two individuals in one new individual. Okay, so the sexual reproduction is based on this, and this is going to be evolution. The other super important step is that in anaphase 2, okay, instead of the chromatids breaking, instead of the chromosomes breaking into two chromatids, each chromatid is going to go to another pole. Okay, but remember that I've said that there's going to be fully detailed presentations about this, so wait a little bit more, but you can start studying from this. Okay, this is the very basic stuff. Obviously it's a super long video, but it has everything, actually everything. Well, we don't have here the protein path explained, but short of that, you have everything that we've seen in the first evaluation of biology. So as always, I hope you like it. I will be there going around the school. Just look for me. I will solve doubts. You know, if you don't see me and you have a doubt, just send me an email and I'll answer as quickly as I can. Short of that, night night, and see you tomorrow in school. Bye.